Hello learners, in our today's class we are going to understand about the structures of the MHC class 1 molecules and MHC class 2 molecules. In my last lecture, I had covered about the basic details of MHC class 1, class 2 and class 3 as well as their functions and how they are playing the role in immune system of our body. Now today I have to discuss about the structures of class 1 molecules and class 2 molecules. So in this video, first of all we are going to cover the class 1 molecules and their structural details which are very important for exams, and secondly we are going to continue with the class 2 molecules and their structural details. So let's start with class 1 molecules and its structure. MHC class 1 molecule consists of 4 subunits: alpha 1, alpha 2, alpha 3 and beta 2. The four subunits are obviously the peptides only, and these subunits together comprise the class 1 molecule. The class 1 molecule consists of majorly the two important chains. One is the alpha chain and one is the beta subunit. The alpha chain is this L-shaped chain which consists of alpha 1 subunit, alpha 2 subunit and alpha 3 subunit, whilst the beta subunit is only composed of one unit, that is beta 2 microglobulin. This beta and the alpha chain are together linked by the non-covalent linkages. The alpha 1 and the alpha 2 subunits of the alpha chain together comprise a cleft, which is the peptide binding cleft. The foreign antigen and its peptide will be bound here in this cleft. The alpha 3 subunit is again very important because once the antigen has bound here, the class 1 molecule has to represent this to CD8 plus cells. Those CD8 plus cells will form the linkage with alpha 3. So here the CD8 plus cells will come and bind. So in short we can say that the cleft is formed by alpha 1 and alpha 2 subunits wherein the antigenic peptide will bind, and the alpha 3 subunit is that part wherein the CD8 plus cells will come and bind to show their effect. The whole of the alpha chain is anchored with the membrane of the cell. Once this MHC class 1 molecule is exposed onto the surface of the cell, this MHC class 1 molecule will look like this. Only there is a one site of attachment through which this whole of the MHC molecule is anchored into the lipid bilayer of the cell, and that is through the alpha 3 subunit. The alpha 3 subunit and the beta 2 microglobulin subunit are very close to the membrane or are in very close proximity to the membrane of the cell. That's why these are referred to as membrane proximal domains. When we look at the microscopic structures and the let's say from the X-ray crystallography and all the structures of the peptides, we come to know that these peptides, which are comprising alpha 3 and beta 2 microglobulin subunits, the peptides are beta sheets, and there are these beta sheets which are arranged in the anti-parallel fashion, and these are then linked together by disulfide linkages. Now if we look at the structure of the immunoglobulins also typically consist of the anti-parallel beta sheets which are linked together by disulfide linkages. Because the alpha 3 and beta 2 or the membrane proximal domains have a very similar structure to what the immunoglobulins have, so we say that the Ig fold structure is found here in these subunits. The same even goes for the class 2 molecules, because there also in class 2 molecules the membrane proximal units have the similar kind of beta sheath arrangements like we find in the immunoglobulins. That's why we say that MHC class 1 and MHC class 2 molecules belong to immunoglobulin superfamily. So both of them belong to immunoglobulin superfamily. That is because of their peptide arrangements, which are very much resembling the immunoglobulin's peptide arrangements. Moving on further, beta 2 microglobulin is never anchored into the lipid bilayer. The only site through which the beta 2 microglobulin can be attached to all of this MHC class 1 is the non-covalent linkages which are between the alpha chains and the beta 2 microglobulin. Please remember that beta 2 microglobulin is a very important component because without this microglobulin, the whole of the functionality of the MHC class 1 won't be retained. There is another important part which we see here, that is the tail. It is most probably the hydrophilic tail which is in the cytoplasm of the affected cell. So if I say the anchoring segment, this is the membrane anchoring segment, and then there is the tail which is there in the cytosol or the cytoplasmic tail. Through this part you can consider that it is like a tree and the tree has its roots. So similarly here the whole of this MHC class 1 has its roots here. If we try to focus upon the cleft here, which are between and which are formed by the alpha 1 and alpha 2, we come to know that here between the two clefts the peptide, the antigenic peptide will come and attach itself. Now the whole of the depth of the cleft is around 11 amino acids. The width of the cleft is around 10 amino acids and the length of the cleft is around 20 amino acids. The antigenic peptide will come and will bind here. The floor of this cleft is made up of, to be very precise, the floor of the cleft is made up of 8 antiparallel beta sheets which are comprising the floor of this cleft of MHC1 molecule. The side walls, which you can clearly observe here, these are made up of alpha helices. But then you can see there are two openings also, means from the two sides the walls are there of alpha helices, and here we can see there is the opening, but no, there is no such opening. This is also actually covered. So the whole of it resembles like a bowl where we have the cavity in center and then all the surrounding ones we have the things. So here the sideways walls are formed by the certain conserved amino acids. So if I want to represent it in a three-dimensional way, I can say that if we are looking at from the top side view, this is the cavity. The cavity is surrounded by four different walls. The two of the walls are made up of alpha chains, while the other two are made up of some of the conserved amino acids. We are looking at from upside. Now the antigen which is there has to come and has to bind here, means it will be like this way. And because it is very small, the cleft is limited, so the maximum amount of or the length of antigen which can bind here is approximately 8 to 10 amino acids, not more than that. So only a very short peptide of antigen can come and bind to the cleft of MHC class 1 molecule. Now let us try to visualize it by rotating it such that the side walls are made up of the conserved amino acids. We are not considering the alpha helices right now. So if we see the side walls, let me draw it here so that it is very clear to you. If we are considering the side walls, suppose this is my 8 anti-parallel beta sheets floor. This side wall by conserved amino acid, this side wall also by conserved amino acids, and then obviously there are some of the alpha helices here also, but I am not showing that right now. Now the antigen which has to come and bind to it has to bind in a way that here and here should be the two amino acids of the antigen, and then in between the entire antigen should be there, like this way. The whole length of antigen should be around 8 to 10 amino acids, not more than that. When I talk about antigen, I am talking about antigenic peptide, not the complete antigen. Now always remember that this part, this and this amino acid of antigen needs to be hydrophobic, because these are those amino acids which have to make the affinity with these conserved amino acids. Here also hydrophobic. So please remember that when we talk about the cleft of class 1 MHC molecule, the antigen which we are trying to show here, the antigenic peptide which we are trying to show here, is linked to the entire cleft only by two important amino acids, that is the first amino acid of that peptide and the last amino acid of that peptide. For this, the first and last amino acids have to definitely be the hydrophobic ones so that they can have this close affinity with the conserved amino acid sequences. Because might be possible that these conserved amino acids are also hydrophobic in nature. So this is a typical class 1 molecule, its cleft, its functioning, and how the peptide, antigenic peptide, binds to it. Please note that in the previous lecture we were talking about the HLA genes, that is HLA-A, B and C. These genes were coding for the MHC class 1 molecules. So now we can look into the specificities that in MHC class 1 molecule only the alpha chain, that is alpha 1 plus alpha 2 plus alpha 3 subunits. Together, the three of these chains, only these three subunits or the whole of alpha chain is only coded by the HLA A, B and C. When I talk about the beta 2 microglobulin, it is coded by some another gene, so on some another chromosome. Let's put it a question mark. So on chromosome 4, the genes which we were talking about in the last lecture, the three genes HLA A, B and C, which code for MHC class 1 molecule, actually are coding for only the alpha chain of that protein or only the alpha chain of that MHC1. The beta 2 microglobulin has to be coded by another gene which is located on some another chromosome. So this is whole of what we are talking about the MHC class 1. Now let's move on to the MHC class 2 structure. So this is how a typical MHC class 2 structure looks like, and it is also referred to as, popularly called as, the dimer of heterodimers. So it means that, so they're popularly referred to as the dimer of the heterodimers. Why they are called so? If you look at the structure of MHC class 2 molecule, it is very much dissimilar to what MHC class 1 looks like. In MHC class 1 we had this kind of structure, but in MHC class 2 we are having this kind of structure. There are two chains, alpha chain and beta chain, altogether. The alpha chain is comprising of alpha 1 and alpha 2 molecules, which means it is a heterodimer. Similarly, the beta chain comprises of beta 1 and beta 2 molecules. It again means it is a heterodimer. And the two heterodimers are forming the dimeric linkages, so it is popularly referred to as the dimer of the heterodimers. Now again, just very similar to class 1 molecules, the MHC class 2 molecules here are also associated with the non-covalent linkages. When I talk about the non-covalent linkages, where can they be found? They can be found in between the two uh, heterodimers. So suppose if you are looking here the peptide, this is the peptide of a beta chain and this is the peptide of alpha chain. So in between them also there might be some non-covalent linkages. Similarly, there might be some of the non-covalent interactions here, which is comprising together of this MHC class 2 molecule. Now one thing which is noteworthy here is that the alpha 2 chain is also anchoring inside the membrane and the beta 2 subunit is also anchoring inside the membrane. So indirectly we can say that whole of the alpha chain anchors into the membrane and whole of the beta chain also anchors into the membrane. Now the anchoring is again done by one of the membrane spanning domains, that is the kind of alpha helix, and then afterwards there is the cytoplasmic tail. The same here also goes. Now let us come on to the cleft of the class 2 molecule. Let us not move on to the dimensions of this cleft, and let us try to cover this in a generalized form. Again, when I talk about the platform of the cleft or when I talk about the base of the cleft, the base of the cleft is comprised of 8, sorry, let me, yes, 8 anti-parallel beta sheets, which is forming the floor of the cleft, and then afterwards there are two side walls which are formed by what? Which are formed by the alpha helices. Then the two remaining walls, two sides, which in the MHC class 1 were covered by the conserved amino acids, these two sides here are open. Now what is the function and what is the advantage of this opening? It is very important to note here that if the two sides are open, that means that the antigenic peptide which has to bind here now has some of the freedom. That means that now the longer antigen peptide can also bind into this so that it can pass through it. Like it is not bounded to a limited space, it is bounded to a narrow tunnel. So in the narrow tunnel now a longer antigen can also be binding here easily. So here the restriction of the antigenic peptide increases to about 13 to 18 amino acids long. While in class 1, if you remember, in class 1 we had only about 8 to 10 amino acids long. Why? Because in class 1 the cleft was covered from all the four sides, but in the class 2 it is not the case. The two sides are open, so there the antigen can easily span through the two open areas. Now you might be thinking that in the class when we talked about that the conserved amino acids which were present there, the antigen was forming some kind of bonds there, the peptide, and hence the antigen was attached here. But what about the class 2 molecules? So in the class 2 molecules, this is noteworthy again that the antigen would not be lying in this way. The antigen is in a way that, suppose these are the two side walls which are formed from the alpha helices and this is the floor. So if this is the case here, and here also the two walls like this way, as simple as that. I should also highlight it so that you can easily understand. It is like this way. The two walls are this way and then there is a floor here. I think now you can easily imagine it, right? So now what is happening is that because from the sideways, let me zoom into this, because here there are no, sorry, uh, yes, because here there are no conserved amino acid sequences, so now there is a full length in which the peptide can be attached here. So it can be approximately 13 to 18 amino acids long which can be attached here. The main point is that now how it should be attached here means what is the linkage. So it is forming the linkages through these alpha helices. So there might be some of the important linkages through which the peptide can bind or peptide can be linked to these alpha helices rather than linking through those amino acids. So this is an important thing and that's why it gives a much more freedom to MHC class 2 molecules so that they can even express the peptides which are little bit longer in length. So please remember the length restrictions for MHC class 1. It is, as I told, it is 8 to 10 amino acids, and for MHC class 2 it is 13 to 18 amino acids. So that is all the structural details of MHC class 1 and MHC class 2 molecules. In the next class, we are going to see how the antigens are presented through the class 1 and class 2 molecules and what are the endogenous and exogenous pathways through which these antigens are represented. In the upcoming videos, we are going to cover the two pathways one by one.